This is Bio 181, Chapter 7 on cellular respiration. And in this video, we're actually talking about fermentation, which is when we have no oxygen. Okay, so oxygen is not used and glucose is only partially oxidized. That means only some of the energy is taken out. So what happens is after glycolysis occurs, where we make 2 ATP, we regenerate this electron carrier NAD plus. Okay, so it's regenerated through a process that creates either lactate, and that is called lactic acid fermentation, or in other organisms, the product is ethanol, which is alcohol, and carbon dioxide gas, and that's called alcoholic fermentation. So you still start off with glucose, and you still go through glycolysis, glycolysis. So we're splitting it in half. And we make, in this process of glycolysis, we make 2 ATP. And that's it. So we make 2 net ATP from glucose. Then we are going to generate pyruvate, okay, but it's not further oxidized. That molecule will then be converted into a byproduct. Glycolysis also generates the electron carrier NADH. Okay, so if you remember, NAD plus is the form that does not have the electrons. Once it has the electrons, so the electrons come off of glucose and they're actually added to NAD plus to create NADH. However, glycolysis cannot continue. It can't keep running unless it has more NAD plus to take these electrons. So fermentation is a way to regenerate. So it regenerates the NAD plus, and it allows glycolysis to keep going. All right. So visually looking at this, again, it's the regeneration of the electron carrier that is required for glycolysis to continue. So this is without oxygen, right? We have pyruvate, which is the byproduct. It's the product of glycolysis. So this is the product of glycolysis, which is the first step in cellular respiration. It occurs in the cytoplasm, glycolysis. And some organisms, that's all they do, right? So they're only going to do this predated aerobic cellular respiration. So you get pyruvate, and then there's a couple options. So the option with oxygen is you go into cellular respiration, and you talk about that in the other videos. We're concerned without oxygen in this video. So pyruvate is coming over here, and it's either going to make ultimately lactate. So this is lactic acid fermentation. This occurs in the muscles of humans. We generate lactic acid. So as soon as we use up our oxygen, which happens pretty quickly in muscle contraction, we switch to anaerobic respiration and we generate this lactic acid. Okay, this has to then go to the liver and then be made into pyruvate. Okay. Again, the whole purpose here is we need to regenerate this molecule NAD plus so that we can continue to do glycolysis. So this is option one. Option one. So some organisms do this. This is what humans do. Okay. Lactic acid fermentation. Option two, I'm just going to put a different color over here. So this is option two, without oxygen still. Okay. In this reaction, the way that we end up getting NAD plus is first we make the byproduct carbon dioxide. You go through an intermediate called acetaldehyde. You don't have to memorize that. But what happens here, importantly, is you regenerate your electron carrier, and then you end up with a byproduct of alcohol. Okay, so ethanol is a type of alcohol. This is the alcohol that we drink and that makes you drunk, right? And so this is called alcohol fermentation. And a lot of alcohol has bubbles in it, right? It's carbonated. So that's the CO2. So you make alcohol and you make carbon dioxide. This happens, organisms like yeast. Okay? If you bake bread, you feed the yeast a little bit of sugar. They eat the sugar to make ATP. As a byproduct, they make CO2, which is why bread rises. Okay, the air bubbles or the CO2 bubbles get stuck in the gluten, the sticky protein in the bread. And then they also make alcohol, but that, if you're making bread, will vaporize so you don't get drunk when you eat bread. Okay, so the two types, fermentation follows glycolysis when oxygen is not present. Okay, so some organisms can switch between using it and not using oxygen. Others are just all one way or another. Okay. But the purpose is to recycle the electron carrier, specifically NAD+. So we can continue using it again in glycolysis. It reduces organic molecules. So that word again, reduction, involves gain, right? So oil rig, oxidation is loss, reduction involves gain. So it reduces organic molecules in order to regenerate NAD+, which means it gives them these electrons. So it's kind of like, wow, you're just giving away this energy, but yeah, it's necessary to keep the process going. So that energy, the electrons either go into ethanol, and carbon dioxide is a byproduct, or those electrons end up going into lactic acid. So you'll see the word lactate or lactic acid, those are basically the same thing. One of them when it ionizes and in water, but lactic acid or lactate. This is just showing the diagram, lactic acid from our book. I just wanted to bring this up so you could recognize it. You start with glucose, you go through glycolysis, you end with pyruvate. You take that pyruvate, you reduce it to lactate, right? So you're adding in the electrons here. They're being added to this molecule, producing lactate. That's our byproduct. Regenerating NADH goes back in, so glycolysis can continue to occur. Alcohol fermentation, a similar process. You take glucose, you go through glycolysis, you make pyruvate. In this process, again, you're making 2 ATP, which is why we do it, why yeast does it. Yeast is eating the sugar in the bread and the flour, and if you give it extra sugar, it's super happy. So yeast is an organism, it's a fungus. It consumes the sugar, it makes ATP so it can live and reproduce, right? And then it creates pyruvate, which it's done with. It doesn't have the ability to get any more energy out of it. So it takes some of the carbon in it, releases as carbon dioxide. The remaining carbon will be converted into alcohol, again, with the addition of extra electrons, creating that alcohol byproduct. So the products of alcohol fermentation, it's easy to remember alcohol because it's in the name, ethanol. One thing people usually forget is the carbon dioxide. All right, so this is the same topic, then I have one more. So aerobic cellular respiration is much more efficient, right? So aerobic cellular respiration making about 32 ATP, whereas fermentation makes about 2 ATP per glucose molecule. So per 1 glucose molecule, right? So with the presence of oxygen, you can make 32. Without it, you only make 2. But 2 is better than nothing. The very last topic is, okay, what about I eat, a whole bunch of fat and protein? How does that get broken down into energy? Well, we're going to start with the catabolism of fat really quickly. If you I thought we were going to do that first, but let's actually not do that. Let's start with protein. Okay, so protein, we're starting with protein. So protein, remember, is made up of amino acids. And so the amino group is removed first. So here is an example of an amino acid. It's got a carboxyl group over here, COOH. It's got the amino group here. Oops. And where are we at? Oh, NH2, right? So the amino group is combined with another hydrogen. It is released and that becomes urea. So urea is the biggest waste product really in our urine. If we are not able to take that out through this process of metabolism, we get pretty sick. All right, quick. So then this deaminated, they remove the amino, it just means now it doesn't have the amino group. Now this is going to go into the citric acid cycle, which is a stage in aerobic cellular respiration. Fats have these huge hydrocarbon chains. They've got tons of carbon hydrogen bonds, right? Just tons of them. So it turns out that, for every 2 of these, it can break this up into here's 2 and here's 2 and here's 2. So it can take this group, this is called an acetyl group, and it just takes it and puts it into, again, aerobic cellular respiration, brings it into the citric acid cycle, and it can be broken down into carbon dioxide and make a little bit of ATP. So for every acetyl group, every 2 carbons, it can get 2 ATP out. So very energy rich. This is the diagram showing that. I won't belabor it. Just want to highlight proteins broken down into amino acids. And then those are taken and put into the citric acid cycle after deamination, which is the removal of the nitrogen. And then for the fats, okay, so for the lipids and fats, breaks it down into fatty acids, brings that in as acetyl-CoA, which then goes into the citric acid cycle. And then through this process, we're getting out ATP. All right, that's it.